Thank you for coming and joining us at the International Webinar Series by IMMB. My name is Dr. Hyo. I am the Director of IMMB. And today I would like to introduce one speaker to all of you. First of all, I welcome all of you to this webinar. All right, Professor Dr. Hyo-Bang is our speaker today, and we are very grateful and thankful that Professor Moon is physically with us in our institute, IMMB, Faculty of Medicine, UITM. Let me introduce Professor Moon. Professor Moon is a professor at the Department of Marine Science and Convergent Engineering, Hanyang University in South Korea. He is an environmental analytical chemist with good knowledge in environmental chemistry, wildlife ecotoxicology, and human exposure to persistent organic pollutants, in short form POP, POPs. And also his expertise is in emerging contaminants. He got a PhD degree from Pukyong National University in 2003. In the past, he had worked for the National Institute of Fisheries Sciences as a research scientist. And since 1999, he has published over 230 peer-reviewed articles. I think, Prof. Moon, you have lost count since then. Right? Too many manuscripts published under his name. Yeah. And his paper mainly related to marine pollution, bioaccumulation, analytical methods development, biomonitoring, and human exposure to POP and emerging contaminants. And Prof. Moon was selected as POPs, POPs Review Committee under the Stockholm Convention of the United Nations Environmental Program, UNEP. And Prof. Moon mentioned to me that he was one of the members who can vote in any decision made by the United Nations in this matter. And also he is serving as the associate editor in Ecotoxicology and Environmental Safety Journal with impact factors of 7, and also Frontier in Marine Science, impact factor 5, and there are many more like Chemosphere, Journal of Emerging Contaminants, to name a few. And to date, Prof. Moon has a hash index over 50 and total citation more than 10,000. All right. And currently, this is one of the latest updates. Prof. Moon is also the president for Korea Persistent Organic Pollutants, in short form, KPOPs. He is the president of KPOPs, right? That's very impressive. And Prof. Moon, last year, he was also won the Hanyang University Outstanding Research Professor Award. So without further ado, I would like to invite Prof. Moon to deliver his talk, which is entitled Paradigm Shift for Environmental and Biological Monitoring with a focus on the OPFR. Yeah, Prof. Moon, the floor is yours.
Okay, thank you. Nice little introduction here. So good to see you, all of you. And also thank you for coming and joining to this international webinar seminar. Today I'm going to talk about the paradigm shift for environmental monitoring and biological monitoring with a focus on the organophosphate flame retardant. Okay, before my presentation, maybe some that I needed to explain about the chemicals, the targeted chemicals, organophosphate flame retardant. So I think those kinds of chemicals can be defined as emerging contaminants or emerging concerns or emerging contaminants. So I want to show you the history of the organophosphate flame retardant. Okay, when you talk about the emerging contaminant, so we can use those kinds of the chemical abstract service registry. So when you do, if you want to find new chemicals, you cannot ask some the chemical range. For example, the H2SO4. We can just suggest some the CAS register number. This is a very unique identifier of the individual chemicals, which means if you track the number of the chemicals in the CAS Chemical Abstract Service database, we can know how many chemicals are consuming in our society. So in 2015, the chemical numbers already exceeded 100 millions. And then I'm not sure, maybe, so I think it's on the double up to almost 200 millions. So based on this time trend of the chemical numbers, every year 5,000 chemicals are registering, and every day 5,000 chemicals are registering, and the chemicals are revealed in our industrial markets. So my question is very simple: which one should be prioritized among all kinds of chemicals? And also, all kinds of chemicals are safe to our maybe our environment, ecology, as well as human. So based on some the epidemiological studies, this is the Korean cases. So we call the environmental disease. So the time trend of BMI in Korea is increasing over time, especially some time trend of precocious poverty in Korea. Yeah, for some the girls, is exponentially increased over time. So, and also, sorry, so I used just the Korean languages. So this is kind of the reproductive diseases over time. All some the environmental diseases increasing. Many scientists are pointing out that those kinds of trend could be associated with the increased amount of chemicals. So the WHO reported some EDCs status report. So they mentioned about the health effect burden of endocrine disrupting chemicals. Yes, so almost caused by health effect. However, so those kinds of statistical suggestion is just the tip of iceberg. It means that we should consume too many the payment, the cost burdens to solve the endocrine disrupting chemical problems. So among them, so when I studied about some the chemical monitoring, I usually use those kinds of division categorization. So the chemicals can be divided into organic and inorganic. Organic chemicals based on their physico-chemical properties, very persistent endocrine disrupting chemical, we call the PAPs. And also less persistent chemical, endocrine disrupting chemical, basically bisphenol A, the phthalate, including DEHP and paraben. And some chemicals are revealed in recent years. We call the emerging contaminants like siloxanes, benzotriazole, UV stabilizers, the synthetic muscat compound. Okay, among those kinds of chemicals, some based on the regulation program in global and domestic scale, so many chemicals are replaced by new chemicals. We call the alternatives or the replacement. For example, PBDEs are regulated by Stockholm Convention. So all the companies to produce electronics could not use some of the PBDEs for their purposes. So novel brominated flame retardant have been introduced. So my concerns on those kinds of alternatives. Today, this is actually some of the coverage of my studies in my laboratory. So today I'm gonna talk about organophosphate flame retardant and their metabolite form of the OPFRs. Okay, so let's beginning some the flame retardant, the understanding the flame retardant. FRs are the additives added to some consumer product to delay ignition. So we can think about the fire event. So we need time to escape from the fire event place. So here, we have some two SOFA. One SOFA is included some the flame retardant. This SOFA did not contain the flame retardant. If you give ignition, so we can see some completely different status for fire event. So we mean just some the flame retardant are using for delaying some ignition when the fire event is occurred. So insulation, textile, furniture, electronics, cable, and baby product, all kinds of some plastic-based product should be included with a flame retardant to prevent a fire event. So the flame retardant can be divided into bromine, chlorine, phosphate, and other inorganic contaminants. So among the flame retardant, 50% of decabd were consumed in Asian countries. Among them, Korea consumed 25% of decabd. It means some the Korean, as you may know, some electrical or electronics companies like Samsung, LG, SK Hynix, those kinds of some global company want to produce many electronics. So we need flame retardant to make those kinds of electronics. So PBDEs, HBCDDs were nominated as the persistent organic pollutant based on their physico-chemical properties and also the environmental characteristics like persistence, bioaccumulation, long-range transport, as well as toxicity. So many industries want to develop new alternatives instead of some regulated chemicals like PBDs, HBCDDs. The alternatives can be divided into types: novel different, chlorinated some flame retardant like Chlorine Plus. On the other one, organophosphate flame retardant. Looking at this figure on the demand of the flame retardant in Korea, the yellow color is brominated flame retardant. Polybrominated diphenyl esters, hexabromocyclododecane is brominated flame retardant. Their consumption are decreasing since 2004. However, the other compound like the small color, maybe wine color, this is a phosphate flame retardant, including OPFR's consumption is increasing over time. So in 2013, the global market, some the organophosphate flame retardant consumption was estimated to be almost 700,000 tons. So it means the high volume, highly consumed in our industry markets. So normally I call the OPFRs as a re-emerging contaminant because this chemical was already synthesized in 1960s. So for the use of the flame retardant as well as plasticizers. So some the researchers studied about the environmental relevance or the toxic effect of these chemicals to the laboratory animals. At that time, especially in 1980s, all studies focused on the persistent chemicals in the environment and ecosystem. So OPFR studies were stopped because these chemicals are not concerned on not so persistent in the environment as well as in the food web. However, since 2000, organophosphate flame retardant become very important chemicals because indoor environment like indoor air and indoor dust were regarded as some very important exposed pathways of the toxic chemicals to humans, and also highly consumed chemicals can be exposed to highly amount to human as well as ecosystem. So organophosphate flame retardant included as the priority chemicals in the European Union, as well as some other countries. So instead of some of the regulation program on polybrominated diphenyl esters and HBCDD, those kind of some of the brominated flame retardant, the regulation, the OPFRs were reintroduced to our society. So I called OPFRs as the emerging contaminant. Okay, looking at the timeline of some of the overall phosphate flame retardant regulation, there are so many the policy and also some action plan and the targeted compound. In summary, all kind of the regulation program was focused on the chlorinated organophosphate flame retardant, TCP, TDCI, I will call so many chemicals, but I could not remember all the chemical abbreviation. Too many OPFRs are consumed and revealed in our society. So first is the child care product, electronics, and furniture. And in Korea, so we have some global companies that they just followed the global regulation program to export our electronics to other countries. Okay, so what's happening to the OPFR contamination in Korea? So environment distribution and the human exposure. So it was surprising because yesterday I visited the shopping mall. So I found some the water purifier, yeah, in the shopping mall, Cuckoo and SK Magic. So I don't know why. So it's very interesting because I published the OPFR in drinking water in 2016. The water research. This is a 1% top journal. So at that time, I analyzed organophosphate flame retardant in drinking water. The reason why, so one company, this is the big company, asked me analyze OPFRs because the two OPFRs were should be removed during drinking water treatment facility, like the filtering system, because they should take some certificate from NSF, National Sanitation Foundation in the United States. So with the certificate, they can export the product to other countries, especially European countries and the North Americans. So they asked me to analyze this chemical, but at that time, nobody analyzed OPFRs. So I do not believe the detection of OPFRs in drinking water, in the water purifier, the water purified waters, because this is a flame retardant. Flame retardant should be used for flammable product. So water purifier can be contacted with water. No need the flame retardant. But I could not believe it. So and also I compared some of the purified waters, bottled water, tap water. Purified water showed us on the highest level. This is the log scale. It's a very stupid story. You want to take safe waters, so we want to use some of the water purifier. But tap water is a relatively low concentration for OPFRs, but fast flows on the water purifier, OPFR, this is a carcinogenic contaminant. The concentration increased. So I was wondering why. So I analyzed some OPFRs in the water purifier part. And I found especially a very extremely higher concentration of OPFRs in O-ring and other some of the part. So I could not believe it because almost all major consumption of the O-ring part is some of the electronics, other electronics. So they need some OPFRs. So water purifier just employed this O-rings without any examination of OPFR detection. So you found very high levels. So in Korea, so the 50% population are using the water purifier every day. So we calculated some human exposure levels of OPFRs extremely high. Yeah, it's almost comparable to indoor dust. And also, my study was expanded to the marine environment. We collected sediment samples and bivalves. The bivalve means the two shells of shellfish. And we analyzed some of the several kinds of OPFRs. We found some of the widespread contamination by OPFRs, especially some of the harbor zones showed some of the highest levels of OPFRs, which means some of the vessels or the ships need some of the flame retardant, for the preventing the fire event on the sea. And also we expanded our study in very specialized coastal zone. This is the Lake Shihwa, which is very close to my campus. This is artificial saltwater lake. So this lake was highly contaminated OPFRs. The title, this paper, the title was some global hotspot of OPFRs in Korean lake. So sometimes it's very proud. I'm very proud of my findings, but sometimes very bad news to me because my house is here. Okay. And also I analyzed I monitored some of the Ulsan Bay, which is a very specialized by petrochemical and very big research, vessel production, and also some non-ferrous flame retardant. Looking at this figure, so polybrominated flame retardant and other novel brominated flame retardant, the contamination pattern is a completely different with organophosphate flame retardant. Here is the Tewa River is here. Unlike the other chemicals, OPFRs were contaminated by some the city zones, not like some the industrial complex zones, which means OPFRs are highly consumed in some the human society compared to industrial complexes. And then my study expanded to the Arctic environment. I visited some the Dasan station in Korean, the Arctic station, and they collected the sludgy samples, seawater, sediment, soil, air, and sea ice. And we analyzed those many chemicals using GCQ-TOF, the time of flight mass spectrometry, which is the approach to see some non-target screening analysis, very, very well advanced technology. And you found some of the organophosphate plant retardant in almost all environmental medium. It means that OPFRs can be transported over long distances, even to Arctic region. Because Arctic region do not produce and do not use some OPFRs, but the chemicals are transported by oceanic current as well as atmospheric transport mechanism. And also indoor environment. So we collected some the indoor the dust. Many other countries, Pakistan, India, Cambodia, Vietnam, China, Kuwait, Romania, Saudi Arabia, Greece, United States, Japan, South Korea, except for Malaysia. So the last month, some elective program students visited my laboratory to with some the Malaysian indoor dust. I think Malaysian dust anywhere may be located some within the ranges. Okay, anyway, the South Korea showed some the highest levels of the OPFRs because we are now using too many OPFRs in our house as well as some industrial complexes. And also you assess some human exposure of the PBDs and other OPFRs compared to this figure that is tiny, but OPFRs showed some the highest levels of contamination. Yes, I can conclude OPFRs should be contaminants of emerging concerns. And also we conducted the human biomonitoring for organophosphate flame retardant with some aid of the Adrian Kopsi, who is some editor of the Environment International. So, and also we found several metabolite of some OPFRs. So it's very surprising because some of the exposed levels of OPFRs in Korean population were the highest among whole populations. So it was very surprising news, and with accumulated evidences on OPFRs for ferrant OPFR contamination or metabolite of OPFRs. So, for example, some the metabolite, when you do human biomonitoring, you are not routinely measure some metabolite of OPFRs. This is a kind of some the principle. Okay, so if you expose the OPFRs by ingestion or thermal exposure, maybe they will be eliminated from our bodies by urine. So if you found any metabolite OPFRs, we can recalculate some of the exposure levels of OPFRs. This is the basic principle for the human biomonitoring. However, I have many doubts on this one. I have many skeptical insight to those kinds of the environmental monitoring and biological monitoring. Okay, because all monitoring programs on the machines you have, So if you have some just a GC mass, okay, I can measure this chemical using GC mass. I have LC mass, okay, I can measure metabolite of OPFRs. So in my laboratory, I have enough machines, some LC top, GCQ top, and LC mass, GC mass, any type of machines I can operate. So up to now, if you do environmental monitoring for water and sediment, normally we can measure the parent compound. Okay, so human biomonitoring, all people want to measure metabolite. This is a kind of the principle. However, so I'm doing some of the wildlife biomonitoring, like birds and marine mammals, seals, whales, dolphins. Sometimes the ferrant, sometimes the degradation product, like metabolite. It's very, very I have many skeptical insight on the human and environmental monitoring. What do you think about the organophosphate flame retardant? Okay, when you do analyze or when you do monitoring the POPs, maybe POPs is very persistent, so you can measure their ferrant form. They are not biodegradable in our bodies. Yes, it's acceptable. And also some the POPs cannot be in urine samples. So you should employ the serum samples or adipose tissue samples to measure POPs because they are not changeable. Yeah, by some biodegradation processes. However, OPFRs, based on physical-chemical properties, this could be located between POPs and the legacy persist endocrine disrupting chemical like phthalate. Phthalate very easily degraded in our bodies. So based on their physical-chemical properties of OPFR, look at the 150, 1500. And also there are Kyoto values from 1 to almost 10. There are wide range of hydrophobicity and water solubility and also molecular weight. I believe some chemicals could not be easily degraded in our body. However, some very persistent chemicals could not be changed in the environment. However, we just keep our principle. When you do environmental monitoring, okay, you should monitor ferrant compound. When you do biomonitoring, okay, you should do ferrant compound. You should do measurement of ferrant, not the metabolite compound. So I want to look for what's happening to OPFR in the environment, ecology as well as human. And then I'd like to suggest which form is the more suitable for our monitoring purposes. Okay, this is the analytical challenges. I think I have some limited time, so this is a LC Anyway, my conclusion is we developed a simultaneous measurement of OPFRs for ferrant and metabolite using LC-MS. Okay, so let me this one. This is the ferrant compound. This is a metabolite. Some metabolite can be the commonly used for different compound. So this is, I think, some kind of the limitation. For example, if you detect TPHP in urine samples, actually you don't know which were originated from TPHP, CDP, EHDPP, TCP. Yeah, because they are the common metabolite in samples. So, but many people are just calculating TPHP was from TPHP. I know so. So those kinds of to overcome these challenges, I developed these chemicals. The OPFRs can be divided into 3: alkyl, aryl chlorinated, and their metabolite. This is a tri-OP is parent compound. Di-OP is metabolite, and the other form is a hydroxylated OP is those kind of chemicals. All the metabolites are not commercially available. Some standard were obtained from my friends' lab, some like Adrian Kobashi lab, and also other people. Sometimes I asked to synthesize some chemicals to measure by the monitoring program. And then we made finally I got the successful story of all contaminants. And some pages established analytical procedures of OPFR, the water sediment. I'm not developing the wildlife species samples like mammals and the bird samples. We used some SP cartridge for measurement of OPFRs. Okay, finally, I'd like to introduce my findings, the occurrence and partitioning of and degradation product of OPs. Okay, so many people are thinking about maybe abiotic samples like water and sediment could be detectable for ferrant compound. However, so the result is not so relevant for ferrant compound. Okay, so the Masan Bay is a very highly contaminated site in Korea. Okay, so this is freezing. Okay. So this is surrounded by industrial complexes, and the wastewater treatment plant outlet is discharged into the bay. So Korean government designated this bay as special management coastal zone. So many people are conducted for toxic chemical monitoring program. So we collected the water and sediment samples from Masan Bay, and also we collected some sediment samples from the outfall of the WWTP with some the triangular lines. This is my student. Okay, this is the first findings. We measured some OPFRs in seawater samples. Unlike my expectation, too many degradation products, tri-OPEs or the hydroxylated OPs were detected in water samples even too. And also parent samples were detected. Their concentration is comparable. Parent compound 116 nanogram per liter, or degradation product is almost 17 nanogram per liter. So the detected pattern is the pattern were matched with the consumption of OPFRs in Korea. For example, TP, TCI, PP, TCP, TBOP were highly consumed. This was the TBOP, TCI, PP, TP, TCEP. It's very interesting. Even though TCEP was detected in water samples as parent, but it was not detected from the degradation product. EHPHP were, this is the metabolite, it was detected in water samples, but the parent compound was not detected. It means that some of the TCEP cannot be changed into metabolite. EHPHP very fast, rapidly changed into transformed forms in the water samples. Okay, looking at the in sediment, a little different with the water samples. For example, TP, TCI, PP, TCP, TBOP, there were looked like some the dominant in the sediment samples. However, THP, TPHP is a very small amount of some of those kinds of chemicals well consumed in Korean industries, but they were highly detected in sediment samples because they are very, very hydrophobic contaminants, very easily adsorbed with the sediment particles. So when you talk about the environmental relevance or environmental monitoring, the water sample is like some snapshot. Okay, so if you use some chemical, water can be easily reflected for their contamination state. We talk about the sediment monitoring, chemical properties. Okay, so however, if you're looking at the degradation product, is very interesting. I will explain about later. Okay, so we looked at the composition profile of OPFRs in water samples and sediment samples. What do you think of this? Water sample, the tissue was dominant in as pH chemical changed into the environment. But you are now still monitoring some those kind of chemicals, this part, the blue part, in environmental samples, abiotic samples. Unlike water samples, the sediment shows some half and half. But we just miss many the information from the parent compound, metabolite compound. So I think it's very about some of those no, it was consumed as a parent compound. Metabolite also consumed as parent compound. When you think about the human biomonitoring, if you measure TNBP in urine samples, maybe it could be from some the TNBP. No, it can be partly derived from the use of direct use of TNBP. Yeah, so this is a very important consideration for OPFR monitoring. And also another concern published in the IMMB, so compound TCP, TD, CI particle element, you should use the compound. But PPP, PD, CIPP, DPHP, kinds of metabolite also very higher toxic, sometimes better than the compound. What do you think about this? So if you monitor environmental monitoring, based just those kinds of parent compound, it's not enough. Maybe we should monitor metabolite together with the parent compound. Okay, I just show you some degradation pathways of a TBOEP. TBOEP water pollution profile, sediment profile. In vivo studies, TBOEP can be changed into several some metabolite, BBEHP and OH-TBOEP. 3H-TBO dominant in sediments, but in water samples, BOEHP were dominant. But you don't know about those kinds of degradation processes in the environment. This is just the in vivo studies. And also TNBP. Okay, so the TNBP can be changed to several, the metabolite. But the 3OH TNBP is some major metabolite of TNBP. However, it was not detected in our samples, or detectable in environmental samples. Unlike this, environmental degradation could be different. So now the organizing processes of the OPFRs, it can be very impactful in our society. Okay. Offline retardants are contaminants of emerging concerns. OPFRs should be simultaneously determined on our monitoring. Studies on degradation pathways OPFRs are required, and the bioaccumulation, biomagnification studies OPFRs are leading. Okay, they cannot endure my presentation. Okay, so another measurement of a contaminant. Monitoring is a whole procedure from study design, measurement of sampling protocol, and measurement of chemicals, quality control, and interpretation of generated data. This is a real complete monitoring program. Okay, thank you for your listening.
Thank you very much, Prof. Moon, for your very impressive and interesting presentation. All right, so now is the time. We have about 15 minutes for question and answer session. So I'd like to open this Q&A session to all our participants who are online. If you have any questions, please feel free to ask questions directly to Prof. Moon. Alternatively, you can also type in your question in the chat box so that I can read out your question to Prof. Moon. All right. Is there any question from our online participant? Okay. Perhaps I can ask the first question to Prof. Moon.
Yep.
So you were showing the organophosphate, the content in the seawater, and also the derivative, the breakdown of the chemical. I wonder what are the factors that contribute to the breakdown of the original chemical in the water.
Okay. When you think about some degradation processes, we have the two processes. The first one is ultraviolet sunlight degradation processes, and the other one is biological degradation process, mostly some microbial degradation processes. So we have so many experience to see some degradation process. Fiber minutes, the chemical concentration graded. Some their half-life under. And also the other one is the microbial process. This is kind of some microbial process. Biological biological. And also this is a very natural process. And also this is a very natural process. And also another one is the synthetic process. Can be we can say like some wastewater treatment. If you wash our body and maybe some body, maybe some the washed water will be gathered into
and some the operator wants to remove some chemical
and some the operator want to remove some chemical,
and then they want to charge the effluent.
And then they want to discharge the effluent. In this process, maybe some sedimentation processes and also some biological treatment processes will
during
be
this process, many chemicals can be
during this process, many chemicals can be
and also some
biodegradation processes will be accelerated during this process.
So is
So two is very
among
important.
them, so I have many
Among them,
on the
I have
microbial degradation
Right.
concern on the
especially
microdegradation process.
in sediment.
Yeah, especially in
On
the
sediment,
sediment.
many some the
On sediment, many some the microdegradation process will occur,
very
and also will be very predominantly sedimentation.
Yeah, that's very interesting, Prof. Moon, because when you mentioned about the microbial in sediments, I assume perhaps the environment is anoxic, which is lack of oxygen. So those bacteria could be the anaerobic bacteria that involved in the breakdown of the chemical. Yeah, that's very interesting. Thank you, Prof. Moon. Is there any question from our participants in the room? If you have any question, please feel free to ask. There's one question online, Professor Moon. In your opinion, this is from No Haslina. In your opinion, if the research want to study on the water quality, is it necessary to collect and analyze data from sediment, or is it enough from the water sample itself?
Okay, thank you for your question.
Okay, thank you for your question. So basically, my opinion is to collect multiple environment matrices together. When you collect the water samples, so the water samples is a final product from inflow from the terrestrial environment, and also action between water and the sediment barriers. So if possible, we can collect the water samples and sediment samples simultaneously. So to me, so I'm a marine chemist as well. So I collected the surface waters and bottom waters and bottom sediment, and different of the layers. The water collection is more reasonable to understand what's happening to the contaminant in the water environment, I think. Yeah, my recommendation is to collect the water and sediment, together.
Okay, thank you, Prof. Moon. Yes, any question? Yeah.
Okay, yeah. Yeah.
So the question from the audience, physical audience here, was you found the organic phosphate compound is more in the filtered water.
Water purifier.
Yeah. What is the next? Yeah. So the question was about has Prof. Moon identified other chemical in the water? Yeah. Are we supposed to be afraid of drinking our filtered water in Malaysia, right? That is a very interesting question. Yeah.
Okay. So your first question about the other chemical measurement for drinking water. Yes, the last year, 2022, Exposure and Health is a very nice journal, top 5% journal. We published the bisphenol analogs in drinking waters. BPA is already regulated by the global and domestic agencies. but other the PPS, PPF, PPAF is can be regarded as emerging contaminant. It can be detectable. Yeah, so actually, so the PPF is the mainly used in Korean drinking water transporting system. So you found this one and publish it. And also some other the POPs like hydrophobic contaminants like PCB and OCP cannot be detectable in water samples. However, the PFAS is very easily detectable in the water samples. Yeah. Okay. The second question about or comment or the opinion some on the safety of the water purifier. Yes. I have many some the doubt on some those kinds of machines. So this machine was made from some part, but the major consumption of this part is the other proposed like some the electronics or tumbler. Tumbler needs some to prevent some the high temperature. Yeah, so they need some the flame retardant, to prevent the fire event. But when you produce some the water purifier without any examination, you just employ some those kind of part, for making water purifier. I think this is a big problem. Yeah. So after then, so this is some the company industry the project. So this company completely changed their water purifiers without any some it means some the OPFR free product. Yeah. So one the company also asked me to develop some the water purifier in terms of safety. So I analyzed so many chemicals using the top mass spectrometry, non-target screen analysis. Too many chemicals were detected. You cannot expect it. So finally, I suggest that you should change some of the plastic line. And also they decided some of the routinely changed in the stainless from plastic to stainless steel. Yeah, so I do not want to open some of the company name, but finally they decided some of which is safer for some the drinking water. I think. Okay, thank you for your question.
Thank you for the answer, Prof. Moon. All right, there's one more question from No Haslina online.
Okay.
She asks about, do you standardize the time of water and sedimentation collection? For example, during low tide, do you standardize the time of collection?
Yeah. Yes. It has some studying. So nationwide monitoring program, we have some the standard operational procedure SOPs. It means some the which date, like some considering the tide of some the sea water, and also, but however, some when you do some the intensive monitoring program, like some the one day, you have to consider about so many things, especially so time and the weather and everything. So our some the strategy is to use some the many students. It means some you want to simultaneously collect the water samples, some different some the coastal zones, including and also some freshwater zone. So we want to collect some the almost the water samples at the same time. This is a kind of a strategy. But the nationwide monitor program, we have some standard operational procedures, to avoid some the mistake, during some the sample collection program.
Okay, thank you, Prof. Moon. Any other questions? Yes.
Sorry.
Because you said that this is the emerging chemical, right? So, is there any international or act on the and also on the affiliate levels of different worldwide or in Korea?
All right. The question is about the OPFR, whether there is any international regulation on OPFR. Yeah, please, Prof. Moon.
Okay, thank you for the question. It's very good.
Okay, thank you for the question.
So
It's very
we have a
good.
regulation
So we
OPFR.
have a regulation on OPFRs, but not for all. So mostly OPFRs. TCEP and TDICPP. So based on some of the toxicity evidences, so it was already well known as the carcinogenic and mutagenic contaminant. So the many industries do not want to use this one, but it was recently due to the regulation on PBDs, so OPFR is reintroduced in our industrial market. So now European countries already banned to use or to produce and the TCEP, TCIPP. In Korea, so major demand for this chemical is electronic or electrical companies like Samsung and LG. So they should export their product to European countries and North American countries. So without any choice, so they should follow some global regulations. Yeah, so both of chemicals were already regulated. However, the other chemicals, the OPFRs were not banned on the use and production in any other countries. So many people are wondering, maybe some of the old regulation program is based on the evidences, toxicity, persistence, bioaccumulation potentials as well. So I think it was not enough to be nominated as regulated chemicals for the remainder of the OPFRs. So one of my tasks is to accumulate some of the strong evidences on the regulation program. Yeah, so we are still going, and also some emerging contaminants should be concerned in our scientific society. Okay, thank you.
Usage of them. Do you think in future there will be more alternative and will the same impact as this?
Yes.
The question is, in the future, would there be any alternative for OPFR, and what would be the effect
I already do your
of the alternative?
So let's think about the PPA. PPA is already regulated. For industry, selected PPS, PPF. PPS toxicity is similar or higher than PPA. If you go to the supermarket, if you can choose some of the PPA-free bottles, water bottles, you should pay more. Yeah, but it can be made from PPF and PPS. It's more toxic. It's very stupid decision. Yeah, this is true. Yeah. So what I want to emphasize is this is we call some of the regulatable alternatives. So this is we call some of the whack-a-mole game. We hit PPA, but another one PPS, PPF. This is very what we are doing now. Yeah, I think some of the OPFR, TCP, PBD is regulated, but new carcinogenic OPFR were introduced. Yeah, I think we need some new paradigm. For example, PFAS as well, PFOS, PFOA, all kind of chemicals, very similar status. Now, some of the regulatory agencies want to change our mind. For example, PPA is problem, toxic. Bisphenols should be regulated. PFOS is a big problem. Okay, fluorinated compounds should be regulated. Yeah. So those kinds of some new paradigm for regulation is some introducing in our society, but so too many some debate between regulatory agencies and industries. For example, siloxanes, silicon. Silicon, some people who study semiconductor, silicon is a perfect chemical, very safe. I do not agree with that. Siloxane is toxic, but without siloxane, without the silicon, I think we cannot survive. Yeah, this computer is also made from the silicon. We know Silicon Valley. Yeah, the computer science, all electronics is silicon. Yeah, yes. I think this is kind of the final directions you should overcome. Yeah, so maybe you have many challenges on those kinds of topics, I think. Yeah. Yes, good point. Basic direction is the less toxic means easily degradable in the environment. But in terms of industry, the less toxic or easily degradable alternatives are not alternatives because they are not effective. So they should consider effectiveness of some the regulated compound. So we have so many now it's some the pending on this issue. I think you're pending.
All right. Is there any more question?
One aspect of yes, it's because effectively was purifier. So I'm just thinking because some of this maybe these techniques or some maybe nothing ordinary, so you people or so I would like to ask when you're working to be able to remove these or to resist the regulation of areas to the metabolite, would it be similar in your opinion what you should say
Yeah, can you repeat the question? Summarize the question.
Okay, so your question is about the safety issue of the water purifier. Again, so yes, we don't know about the full procedure or full processes of some chemicals treatment. This is so because all companies do not want to open their original secret. This is a big problem. And also the next one, the other one is so now the approval system is only for selected chemicals. For example, the OPFR in Korea we don't have any approval system for water purifier, but the global company want to sell their product to other countries, so they need a certificate. So NSF has the two chemicals approval system. So one of my contributions to those kinds of activity is to expand some targeted chemicals with strong evidences, like this chemical is very dangerous, and you found this chemical as well. But all the approval system is focused on, as you mentioned, some targeted parent compound. If some the metabolite of targeted chemicals is very dangerous, you should include this one. It's very important point. I believe almost all chemicals could be removed during the water purifier system. However, always we have some exceptions. Some chemicals could not be removed in the water purifier system. So if you found those kinds of the result, I'd like to do some often this kind of the finding to throw some the international journal. This is a role of the scientist.
So in your opinion, is it better for us to use the purified water or maybe the boiled, right?
Okay, so this is another one. In the paper published in last year, the boiled water is not so safe. Okay, so we compared the tap water and the boiled water. And the boiled water types are divided into several types. First one is after boiled, and we didn't open the lid of the bottle, the jar. And then the boiled water, we opened the lid. We opened the lid, many chemicals in the environment are condensed into the water again. It means sometimes the boiled water safe or not. Before this issue, maybe how can you boil, and then after boiled, how can you treat the water? It can make another situation. I think so. We need more information about this. Our decision.
Okay, thank you very much, Prof. Moon, for a very again very interesting question and answer session. All right, so due to time, we have to close this webinar right now. So again, once again, thank you very much for your participation, and thanks, Prof. Moon, for your interesting talk. So thank you very much, everyone, and have a nice day. Thank you, and bye-bye.
Thank you. Bye-bye.